All right, good morning. We will begin since it's 11:01, and I know other people are joining us as I speak, but I will begin. So my name is Jana Hexter. I'm with the Northeastern IPM Center based at Cornell University in upstate New York, but we serve the entire Northeast region. And it is a last of the warm weather is today. We're meant to get cold weather and winter starting tomorrow. So thank you for joining us. Um, so I am delighted that our topic today is genetics and breeding of mite-resistant and honeybee stocks, and I want to share a few things before we get going. This is being transcribed, so you should have the transcription option show up on your screen. It is also being recorded, and I will send you a copy of the recording once it is done. We have a lot of people who are registered. We have over 350 people who are registered. So I expect that we'll get quite a lot of questions, and the best way for us to be able to handle that is for you not to put the questions in the chat, but to put them in the Q&A. So if you click on the control bar for Zoom, you should have an option that says Q&A. And if you can put your questions in there, it helps me to keep track of what's been answered and what questions still remain. And also, if there are any duplicates, I can kind of see that and make sure that I ask the most popular question. You can also ask questions anonymously too, so please feel free to do that. So we have some questions for you before we begin. It gives us a sense, it's just 5 short questions, and it should be popping up on your screen now. And it gives us a sense of who is on the call and what kind of level of background that you have, because that helps Hong Mei in her presentation. So I'll just be quiet for a couple of minutes to give people a chance to go through. All right, so I will share the results with you. Here we go. There we go. All right, so as level of knowledge of bee breeding and IPM, we have most people falling into the somewhat or not at all knowledgeable, and a slight majority of people have heard about non-chemical treatments for mite populations. And it's about 50-50 whether people have seen varroa mite biting behavior before today. And most people are not sure whether feral bees bite mites or do grooming behavior more than managed bees. And 50-50 also on having heard about land-grant institutions. So hopefully that'll be helpful for Hong Mei and understanding who is on the call. All right. So I'm delighted to welcome Dr. Hong Mei Lee Byerly. She's a fellow of the Royal Entomological Society and a president of the International Branch of the Entomological Society of America. In 2017, she started her research lab on bee genetics, genomics, pollinators, and ecology. She's mentored 40 undergraduate students at CSU and published 49 peer-reviewed papers and book chapters. And she has presented 120 talks for research. So we are absolutely delighted that she is joining us here today. So I will stop my share, and Dr. Lee Byerly is going to then share her slides. So
Great. Yeah. Thank you so much, Jana. It is really truly an honor to be invited to give a talk for the Northeastern IPM Center. I'm really thrilled to see so many uh, audience here. So thank you so much for your Wednesday morning. I'm located in Ohio right now, Central Ohio. So it's misty, raining, cold day. But yeah, hopefully I'll provide some good information to for you to learn about the honeybee genetics and breeding at our university and institution. Excuse me. So I think I would like to start to talk about our institution with some information. As Yana mentioned earlier, I'm at Central State University. So I started my research lab back in August 1st, 2017. And we are the newest 1890 land grant institution. So on the map, you can see on the slide on the right side, there are 19 1890 institutions supported by the USDA NIFA, National Institute for Food and Agriculture. And majority of them are in the southern part of the US. Luckily, we're in Ohio, we're kind of towards the north side. But yeah, we gained our status. And we are very thankful for the President Obama's administration for the giving us the status. So because we have some funding support from USDA NIFA, we are really we aim for provided training and career development for a lot of Black students because our university is a historically Black college and university. So on the left is it's a tower, bell tower of our campus. And the reason I list this is because in 1970s, I think in 1973, if I remember correctly, we had a horrible, horrible tornado. It just came and pretty much wiped out everything in the campus, but our bell tower was standing as the one of very few buildings. So that's our icon for standing. And we advocate for our Black student education, for minority education. And so just start to want to give you some background of my research. I started my research in honeybees back in 2020. No, 2010, sorry. Yeah, so I received my PhD from entomology, Department of Entomology at Purdue University. And I then started doing my postdoctoral research with Dr. Gene Robinson at University of Illinois, Urbana-Champaign. And I'm very interested in studying genetics of insects. And the reason why I started work on honeybees, because I think honeybees are very fascinating. Honeybee model system is one of the best to study both genetics and behavior of animal kingdom. So the bottom here, I just show you a video. This is a pretty common behavior. If a lot of you are doing beekeeping or know about the honeybee behavior. So in a colony, there's one queen. There could be with a standard, a big colony, there could be 50,000 honeybee workers. So the video here is showing you a one worker of a honeybee, most likely a forager, and she's doing waggle dance. And this waggle dance with a waggle run and turn around, it's like a number 8 shape, but it's deliver a lot of information. It tells the other worker bees where to find a flower and nectar sources and how far it is and what direction they can go. So for my research, particularly since my postdoctoral research at Illinois and then later at North Carolina State, and then when I started my own lab, I'm centered in this topic to study the molecular mechanisms and genetics underlying the innate behavior. And based on the biology, we know how the behavior are modulated more or less by the brain, and that's encoded in the DNA of the brain. So that's why I'm pretty interested in the behavioral genetics and epigenetics. So in my lab, we study several variety of topics based on the honeybee model, and including the genomics, the brain and behavior, and stress and physiology. So with the limited time I have today, I would like to to tell you something about the genomics, genetics, and breeding of the honeybees. And you're welcome to check out my other scientific publications in Google Scholar. So let me know if you have any questions. So we'll just dive into the behavior I want to talk about today. So from the title, you probably know I'm going to talk about grooming. And grooming is one type of hygienic behavior that honeybees have to clean themselves, also to keep their hives clean, uh, away or minimize the infestations from the parasitic mice, from pathogens, etc. So from previous literature and a lot of other scientists research, we know the hygienic behavior and high hygienic, high grooming can bring colony with a better health. So when I started in Ohio at Central State, we team up with the Ohio beekeepers. We're interested in this applied question. Can we detect more colonies with a high grooming and mite biting behavior? So this video here I'm gonna play, you will see what the mite biting behavior look like. And this is a type of grooming, which means the worker bee can detect the mite and grab it really quickly, use their mandibles to chew the mite. And a lot of times the mite lose a leg or one or a few, and they may drop down to the bottom board of the colony. And so that's a one way we try to use uh, to score the colony, whether they have high mite biting or not. This is another video. Both videos are recorded by Price Smith and Dwight Wells, and they're both, they were also both Ohio colonies here. So this video, we have a live mite just placed on the top of the frame. And you see a worker bee walk by. And then the mite just grab her. And she instantly, fell all or something on my tummy and started grab the mite and chew it. And now other workers come near here. They also try to help her to grab the mite. So hopefully with these two videos, you can see the mite biting behavior as a part of a grooming. So because we know there are several different kind of feral colonies in near central Ohio, southern Ohio. We want to collect these feral colonies. So these feral bees are wild honeybees. You probably wonder, well, are they just managed honeybee colonies and, got into the trees? So because we team up with the beekeepers, we know they have monitored these colonies for many years. So based on their experience, these feral colonies are located in remote area with a lot of trees and prairie area. So we know there's no other houses or residential houses near them. So most likely these are the feral colonies with living in, untethered in the wild for a long time. And they're not just the package or human managed colonies swarmed from a year before. So we compare these feral colonies. For example, this is one picture of a feral colony in a catalpa tree. I think catalpa tree or walnut tree in central Ohio. And then on the right side, we want to compare them to the control on the right side, which are package bees. So a lot of beekeepers purchase package bees. These are 3 pounds of bees put into a mashed box, and they were signed with a random queen. A lot of these colonies are shipped from southern part of the U.S., for example, Florida or Georgia, at the beginning of the year. So a lot of people order these package bees and they come in April-ish, right? April, May. And then they can use these bees. So because package bees are common in Ohio, so we want to compare them and whether there's any difference of this mite biting behavior between feral and package bees. So the picture here in the middle top is my student. Jada Smith. Jada was my first research student, very outstanding student. She worked with me to go to different apiaries in Ohio. We have collected a lot of mites from the bottom board. The map here show you A, B, and C, 3 different locations. On the right side is the Ohio map. So we have some of the package B colonies or commercial colony from Ohio. And then B site is Central State University site. We're in the Greene County, Ohio. And the C site is the West Lafayette, Indiana. So we're very thankful to have a collaboration with Purdue University. It's Dr. Brock, Professor and Dr. Brock Harper's lab, and also his lab member, Crispin Given. So with all their help, we collected mite biter bees from Purdue. So give you some background information, Purdue University also has devoted many years of efforts now. It's more than a decade now of breeding efforts into the high biting and high grooming colonies as well. So when I started in Ohio, plus I'm also a graduate from Purdue. I always want to go back. So we collaborated and collect bees to compare the mite biting. So what we did is to collect all the mites from the bottom boards and place them on the microscopic slides and label each mite and take pictures of each mite, just thousands and thousands of pictures. My student here is showing my student take the picture, yeah, on the microscope. What do we see under the microscope? So we have a variety of mites. These are all Varroa destructor parasitic mites. The A panel is showing the undamaged. There's no damage of these mice. They have all 8 legs, 4 legs on the left and then 4 legs on the right. The B picture is showing you the mite with the damage, because this mite lost 2 legs now, and there are 2 legs on the left. The yellow arrows pointed out. C and D are the other types of mice we can see. They're probably either not matured or they're too dried out, and we are not sure how these mice are damaged because we don't have evidence of their like legs missing. They could have a whole shell emptied. So it could be from very long time exposure or dropped into bottom board. So from these different conditions, we decided that we only collect the fresh dropped mite. That way we avoid the C and D type of situations. And our intention is to see most A or B type, whether they're damaged or not damaged. So the fresh dropped mice are typically just within 2 days. I think within 2 to 3 days. We clean the bottom board the first day and then wait for 2 to 3 days and we collect the mites again. So that way we get our fresh mites. So we compare first we compare the damaged mites out of the total mites from different colonies. So we have the commercial colony. So the COMM is commercial package bee colonies at the y-axis. We have mite biters from Purdue, that's West Lafayette, Indiana. In addition, we also have mite biters as queen. They're mite biter virgin queen, and they were open mated in Ohio by the Red Patterson Air Force Base. So because the Air Force Base has a huge prairie, it's a Hoffman prairie, which is really big with a lot of flowers, foraging resources. So we know some of the colonies there are also feral colonies of Ohio. That's why, yeah, we have this population of the mite biter open mated. And then, so we compare all these three different population, and we want to compare how many damaged mites versus how many total mites. The data is showing you it's transformed, so it's not directly the strict percentage, but it's after the transformation, because it's a percentage data. So we can see the transformed data. We can see the mite biter and the mite biter open mated are significantly higher with the damaged mites population. In addition, we look at the difference of the average number of legs missing per mite. We see a trend of the mite biter open mated one from Ohio, from the Air Force Base. It's become higher than the commercial one, but statistically it's not significant. So we continue to collect data now. I just don't have the really updated data now. I just show you the data we have from last paper we published. In addition, we also compare the commercial package colonies to the Ohio feral colonies. So for these colonies, we first compare the average number of matured female mites from each colony, and we see there's no significantly different there. And then we also, in addition, added the feral colonies from Kentucky. So this is a collaboration with our master beekeepers, Larry Young, and also our master beekeeper in Kentucky, Dorothy Morgan. So with their help, we can collect all the mites from these colonies. And our data showed throughout the years since 2020 to 2023, these are 4 years of data. We look at the damaged mites versus total mites, and we see a significant difference here. And then when we look at the damaged mites versus the total mites, yeah, so on the left is showing you the significance. The commercial one has the lowest damage. Again, this is transformed data. The Kentucky feral colony, so from the population genetics data that Brock Harper's lab did, so far we know the Kentucky feral colonies are Apis mellifera mellifera. And then compared to the Ohio feral colonies, most likely Apis mellifera linguistica. Both of them, these both feral colony populations have a significantly higher mite biting behavior compared to the commercial bees. And because the video I showed you earlier, you can tell the honeybee worker use the mandibles to chew the mite. So we want to dig deeper into the mandibles, like how do they chew the mite, and will be any difference in their mandibles. So the picture here, the picture on the right just showing you the brain of the Apis mellifera linguistica, and then the mandible is on the bottom here. So almost like the shape looks like a ice cream scoop. So if you eat ice cream, you scoop the ice cream out. So next I'll show you a video here. This is a 3D video. We brought our bee samples into Ohio State University. We use a head scan machine to do micro CT scanning. So let's watch the video. Yeah, so we're thankful with the help from the technician Carly Goodwin to put the video together. So all these yellow highlighted is the one worker bee mandible. Now there's still the same pair of mandibles. We change the color. We try to zoom in now. to look at the really fine structure. So you can see a lot of hair. You see this ice cream scoop shape of mandible. And when we look at inside, so on the scoop part, we can see, so there's a top, this is short edge. I think, can you see my cursor? Yeah.
Yes, we can.
Oh,
Yes, we
okay,
can.
yeah. So you can see my cursor. Yeah, so this is the long edge of the scoop. This is a short edge. So there's also these interesting hooks within the middle of the mandible, and then these kind of a curved shape. So with the micro CT scanning, we're able to compare, so we're able to measure all these different parameters of mandibles. Like, for example, I mentioned short edge and long edge. The W indicates the width. The S indicate the span of the hooks area. The H indicate the height. So with the three-dimensional, we also have a L for the length of the mandibles. So quite interestingly, when we compare these different samples, we have minimum 6 bees per group. We compared a commercial colony versus mite biters. Um, I- and so for this group, I think we have more than 6. I think I'm trying to remember. So we do pairs of each bee. We have at least 3 colonies, and each colony we have at least 6 bees from each colony. So that's, yeah, that'll give at least 18. And the mite biters from Ohio, feral colonies, we can see, no, sorry, this is the mite biters from Purdue University, from West Lafayette, Indiana. We see a significant difference here that the commercial package bees have a higher number of the long edge. So what that indicate is the commercial package bees have a little bit longer on the long edge here. They're bigger than the mite biters. Hopefully that makes sense.
Yeah.
Uh,
So Hung Mei, we have a couple of questions.
oh
So,
yeah.
so it'd be great to just take a break for a minute and ask a couple of questions because
Yeah.
okay. So Jessica Glover said, I've noticed that bees grab and remove small hive beetle larvae if I expose them during an inspection. Is the biting behavior specific to varroa mites or behavior against all pests?
Yeah, so the biting behavior is being used against all the pests. From the papers published, we know the worker bees can use mandible. They can also bite the larva of wax moths. They can bite the mites. They can attack the small hive beetles. But the thing is, the small hive beetle is kind of big compared to the varroa mite. And so for honeybee workers, they cannot really like bite the beetle with a really significant damage there. But as far as I heard from beekeepers or also other researchers, if there's a strong colony, yeah, they tend to kind of, dispel the small hive beetles and try to get them out of the hive when they have a lot of workforce.
Yeah, great. And for folks who may have missed the
Oh, I
beginning,
forgot to mention, sorry. So
yeah.
in addition, not just the biting, the mandible can secrete chemicals. It's called the 2-HT. It can anesthetize the pest. So it can anesthetize the mite, anesthetize the larva of wax moth.
Oh, interesting. So for folks who may have missed the beginning, if you have a question, please put it in the Q&A rather than in the chat. It's easier for us to keep track. And I have another question here from Selena Bruckner. In the figures comparing commercial package bees, mite biters and mite biters open mated, did you pull the data over the 4 data collection years? And if so, did you have to replace queens in the experimental colonies? And how did you deal with the change?
So yeah, so we want to avoid the old kind of empty shell mite type. So we only do fresh job. So fresh job is typically 2 to 3 days. We're looking at 48 hour or 72 hour of the mite job. We don't need to replace queens. So the colony has every colony we tested has a queen in the colony. If something happened, the colony doesn't have a queen, then we will not use that colony. So we want to make sure they're queen right. And I think her question, she probably asking if it's a low biting colony, what do you do? Is that right? Like, do you replace the queen? Because we're doing the like genetics research, so we want to keep the low biting colony as it is. Also, try to see like the high biting colony as it is. So yeah, so we try to not replace the queen
Okay.
in that way. Yeah.
Okay. So we have more questions than we can answer in this question break, but I'll take one more, and it's actually kind of several questions, so I'll let you decide where to go with it. Anne Lichten-Wallner said, in feral bees in general, are varroa mites as big of a problem as in commercial bees? Is the biting behavior, whoops, today's it moved. Is the biting behavior only one defense mechanism against varroa mites, and also are varroa mites present in bumblebees? So there's kind of three questions put in there, so I'll let you decide.
Yeah, that's a very good question. I mean, Aurora might. So I forgot to mention at the beginning, the grooming, my biting behavior is one of the strategies that the colonies have. So the worker bees have a variety of defensive behavior. For example, besides the grooming and removing the mice, they can also detect the brood. So they can detect the sick brood. They can uncap the sick brood and pull out the sick pupa cell from the cells. And then so they could have a high uncapping behavior. There are a lot of research going into that as well. And then also in addition, I think the different populations, they also may have different sizes in the cells. That's another strategy. So think about if the cell, it's a smaller one, maybe less space for the varroa mite to hide in that way. So there are definitely, yeah, a variety of different strategies and behavior against varroa mite. For the bumblebee question, I'm not sure. I'm not an expert in bumblebee. If the bumblebees have varroa mite, this is like first time I heard the bumblebees may have a varroa mite problem. Um, so yeah, but
Okay,
I'll be happy to, read more in the with the literature and try to find out how to help the bumblebees as well.
great. And then there are some more questions about the study, but I think we should carry on with your presentation because they may get answered as we go through. So, and we'll have time at the end for answering more questions. So.
Okay, yeah. Yeah, well, thank you so much. I really appreciate all the questions. That means you're listening to my talk. You're paying attention to my research. Thank you so much. Yeah, so in addition, we'll continue to talk about the mandibles. And these are the picture of two adorable worker bees. The one on the left is Apis cerana. That's Eastern honeybees, or called Asian honeybees. The on the right is Apis mellifera. That's all the honeybees we have in the US. So the Varroa destructor is the infest the Apis cerana long time ago, before they got into Apis mellifera population, the species. So with the natural selection, the Apis cerana by now are mite resistant. They have a resistance to varroa mite now. So for example, when I go to Asia, go to China, they don't have a big problem with the varroa mite. But they do have other problems such as the tropholepisis, the smaller mite problem they have there. So I thought it would be interesting just to compare what's the mandibles look like between these two species, because Serrana has mite resistance now and Mellifera does not have a really effective way for mite resistance yet. That's why we're working on this. So on the right picture, this is phylogenetic tree of all the different species of Apis. And by now there may be a couple more new ones with the always update on the taxonomy, but you can see the Apis mellifera on the top and the Apis cerana is kind of in the middle there. And so the one paper from the Raffadine and Crozier, so they have compared the mandibles of these two species before. And we also know from back in the 1990s, there was also a paper from famous bee researcher Rutner or Runner. His paper also compared the both species, and he didn't find a significant difference. But I thought from, these are 20, 30 years ago of research, maybe now we can look at them again. So what we did is I collected all these different colonies. So in Ohio, with my students, we collected Apis mellifera colony from the Faro colonies. I went to Puerto Rico, so with the help from University of Puerto Rico, Dr. Tuguru Jure, and we collected Puerto Rican bees. The reason for that is because these are used to be Apis mellifera scutellata that are extremely aggressive. However, since the 1990s on the island of Puerto Rico, these bees turn to gentle, and they also tend to have a high grooming behavior as well. So we want to collect these gentle scutellata bees and compare them on the mandibles. In addition, I went to the apiculture, the Institute for Apiculture Research at Chinese Academy of Agriculture Science in Beijing, China. So we collected Apis cerana bees there to compare their mandibles as well. So with these different populations, we have 16 scanned from Apis cerana and 14 scanned from the gentle scutellata bees from Puerto Rico, and then 36 bees from Ohio, Faro bees, and then 10 from the packaged commercial bees. Again, we use a micro CT scanning at Ohio State. We compared all these different parameters. And interestingly, again, we see the same trend like we saw at the comparison of the Purdue bees. The long edge is different. The packaged commercial bees definitely has the highest number of long edge, and all the other bees actually have a lower number, a significantly lower. In addition to the long edge, we also detect the significant difference of this short edge. So you can think of the ice cream scoop. These are the two edges when you scoop the ice cream. And we can see again, significantly lower in the Apis cerana, the gentle scutellata bees, the Ohio Faro bees, but it's higher in the packaged and commercial bees. So this paper, it just got accepted in Apidology and probably gonna come out just in a few days or so, or a few weeks. So hopefully if you have access to Apidology, you'll be able to read more details. In addition, my postdoc, Catherine Farrell, has worked on the transcriptomics. Because we're interested in breeding, we wanted to select the potential good gene markers for selection and for breeding stocks. So we sequenced, we pulled 15 pairs of mandibles from each population. We have a high biting population from Purdue and a low biting from Purdue. And then we also have the commercial population and Faro population from Ohio. What we compared is the 1-day-old bee mandible versus the 8-day-old mandibles. The reason for that is because the 8-day-old honeybee worker display the highest grooming behavior in the colonies. So we want to compare when it's a really young 1 day old versus the 8 day old, what are the changes of their genes. So on the left graph here, the data graph, you will see the C1 for a commercial colony, F1 for feral colony of Ohio. The LB1 is a low biting, one for 1 day old. So HB1 is a high biting 1 day old. When we compare the different, like on the differentially gene expressed, we call it DEG list. And we see the several olfactory binding proteins appeared on the list. So the yellow one is a marker, that's the LV, to show the neuronal cells of the brain. The gray one is olfactory binding protein 6. The orange one, olfactory binding protein 2, and the blue one is binding protein 1. So quite interestingly, we see the high biting colonies, the worker bees 1 day old, they have pretty high expression on the olfactory binding protein 1 in there compared to others. In addition, there's also the OBP2, OBP6. When we look at the 8 day old to compare, we can see the low biting versus high biting one, we can see OBP 1 and 2 and 6 are again much higher than the low biting. And then, but in the feral versus commercial colony, it's kind of a opposite trend. So, which is interesting. But more or less, we have this data to show that not just only the antenna of the honeybee worker, but also in mandibles, we can detect olfactory binding proteins. That means the mandible may also have a function to detect the smell and olfactory cue in chemical ecology and to help on their grooming behavior. So we're still in the middle of analyzing this data. So these could be just primary data and not finalized yet. Yeah. And yeah, because our lab do a lot of sequencing and study genomics and epigenomics. So if you're interested in more information, just please let me know. So in summary, we study the defensive mechanism of the mite biting behavior, and we think there's a link between the shape and the anatomy of the mandibles and their and the mite biting behavior. We also to show there's an evolutionary change now. The trend is that all these mite biting behavior, the colonies with a high mite biting behavior tend to have a smaller mandibles. So you can think of two of the ice cream scoop together. They can cut together, maybe like a Caesar type. But on their short edge and long edge, they tend to be smaller now. So I think we try to explain or justify this smaller size, maybe give them more flexibility in attacking the mites in a way. So we'll make it more flexible. Just one more point I forgot to mention. In general, the Apis cerana eastern honeybees are actually smaller size. Their body size overall, they're smaller size compared to the Apis mellifera. In addition, our goal is to identify more genetic markers and reliable markers for the high grooming and mite biting behavior. In addition, this is the IPM Center. I want to give you a little just one project which just started last year, and we don't have a lot of data yet, but we're working on the experiment. And the idea behind this is to look at a sustainable and non-chemical tool for mite control. I know a lot of people use, miticides, oxalic acid, the check a lot of chemical controls. So in addition, we wanted to explore the non-chemical and perhaps using physical limitations to control the mite population. So the target here is the brood breaks in the honeybee colonies, especially in the fall of the year, because in the fall the honeybee colony decline, the mite colony population will go up, and then they will go down as well. If we can use that time break to target the mites, then that may be very useful. So we aim to interrupt the brood production in the fall, because if we think about the mite reproduction, each female mite has to find a cell, then go in. They can feed on the pupa, and they can reproduce. So what if we don't have a lot of brood for these mites to go into? Then they cannot reproduce. That will disrupt the female mite's reproductive cycle. So the breaking the brood cycle of the mites may help to manage the mites, and this is actually a very standard, typical standard way to do mite control in China. I know a lot of beekeepers in China, in the fall they just cage their queens for 3 weeks. There's no brood, and those mice have nowhere to go and lay eggs. So that's a hypothesis we want to test here in Ohio. We want to cage the queen of the colony, and we want to see if that will decrease the efficacy, increase the efficacy of the varroa control, and decrease the mite population. And because it's a physical way, we don't use chemical treatment, then we have a less worry on the resistance of the mite, on the mites resistance to miticides. So in 2022, my technician, this is Mr. Austin Carey, he worked with students to set up an experiment. We have 3 colonies to cage the queen. We have 3 colonies to have a treatment of Formic Pro, and then we have 3 colonies of a control. And we do the initial mite wash, and we detect, for example, like 10, and 6 mites in 3 different populations. And then we do a final wash after 3 weeks. So you can see the mite, it just increased a lot in the population. However, we didn't see significant difference from the caged queen population. So I think there are variation of the mites in these colonies. So in 2023, this is my other technician, Mrs. Danielle Crow, work with student Kara Allen Digg. We also set up more hives, and so right now we're trying to analyze the data for these. But I think it's not as simple as we thought, just cage the queen and going into the experiment. So we try to optimize the experiments now on the design. We also do selection and breeding program with master beekeepers. So we have Dorothy Morgan come to our campus every spring to do train us to do the instrumental insemination. Our goal is to establish a line. I gave a name, it's Ohio Mite Biter first generation, so OB1 strains, and at Central State. And we also want to maintain high biting colonies as well as low biting colonies, so we can do genetic experiments for the future. So these are some of the pictures. We hold workshops for beekeepers. They're from Ohio, Kentucky, Indiana, and majority of them are just from Ohio. And so before COVID, we had workshop, and then we had field days to open the hives and look at the bees and making the queens. So now we're pretty much back to normal. Our typical workshop days are just end of May or beginning of June. This is our 2022. We team up with the Harlan Honeybee Breeding Co-op and did the supported, and this is supported by the USDA SARA grant. So we train a lot of students on beekeeping, on queen rearing, and bee genetics. These are just variety of pictures with our collaborators, our students, our beekeepers. They're wonderful people to work with. And this is 2023. We hold queen grafting classes to train beekeepers on how to graft the queens. If they see any good genetics or trait of their own colony, then they learn a way how to graft those queens and, maintain and improve the genetics in that way. So these are the training. We have summer camps. We have students. We have summer internships. So these are the variety of pictures, and these are the reference for the talk. And we're really thankful. We want to acknowledge all the funding from USDA. A lot of funding from USDA NIFA, Evans Allen. That's for 1890 land grant, and then USDA SARA grant, the USDA AFRI grant, and capacity building grant, and also our new USDA Next Gen grant for student training. So yeah, thank you so much for your attention. I'm happy to answer more questions.
Great, wonderful. And we do have lots of questions. On timing for folks, just so you know, we're going to take about 10 minutes to answer some questions. And so we will probably finish about 1:15 maybe. It will be recorded for people who need to leave in 5 minutes for another meeting. So a question that we have from Mark Bittinger is how much intra-colony variation in mandible versus the difference across strains?
Yeah, I think that's a really good question. So to answer that, we need to compare the variation within the colony and also across the different strands. We're working on that now. So the example I give for the pooled the transcriptomic data, we put like 15 pairs of mandibles from one population and then compare it to another population. So that'll show the difference across strains. But yeah, but we're not sure the intro, within the colony variation yet.
Yep, great. And Jordan Bethea said, can you share the name of the enzyme secreted by the mandibles?
It's not an enzyme, it's a chemical.
Uh-huh.
So I think it's the 2-HT. I'm trying to.
Yeah, no, this is great because Kathy Misko actually asked a question, which is I was wondering that she asked where
It's
2-HT.
from the mandibular gland.
Okay, great. So she said, is it in the saliva? All right, great. Well, that's answered two questions in one there. Um, Ryan Williamson asked did you track other hygienes such as uncapping or VSH in the three groups?
Yeah, so we start to do that now. In addition to grooming, we start to do the ability of the hive, the workers detect the sick brood. We have UBO kits. I don't know if you heard of it. It's a new kit developed by Kara Wagner from University of North Carolina, Greensboro. So I team up with her company. Basically, we have the UBO kits. We can use the UBO kit. You spray this liquid into the brood, can trick the colony to think these brood are sick. And so then within 2 hours, after 2 hours, you go back to that colony and you can look at how many of them are empty. So, yeah, so that's the one way to detect their uncapping. ability, and that's one we also want to detect. So in the past, the other people have used, for example, the liquid nitrogen. They can do the PVC pipe, and they will go into the brood and use liquid nitrogen. And then within a day, within 24 hours, you can go in to look at how many are empty in that way. The UBO just make it faster, right? So you just wait for 2 hours to do it, and it's a similar way. And then another way people have done before is to use a pain. You just pain kill a bunch of brood in there, but it's very time consuming. So all these are different ways to detect, yeah, hygienic behavior.
Great. And I just had a note from our evaluator asking if we could run the poll that comes at
Oh
the end.
yeah,
So we'll do that
definitely.
and then come back to the questions because I know a lot of people drop off at the top of the hour. So if the poll can show up, it should, there we go. If folks could answer these, this would be great and be helpful for us in knowing the effectiveness of this presentation. And we'll just be quiet so you can concentrate on that. So, all right, I am going to end the poll. There we go. I'll share the results. There we go. So we have an increase in people feeling they are moderately very knowledgeable. And yes, people now understand the question about feral mites and commercial mites and understand more about 1890s and moderately very and somewhat more likely to use IPM. So lovely. Thank you for taking the time to do that. So we have some questions left. So let's see which ones we could do. Um, um, so actually, two people asked a similar question. Michael Lund and Jack Grimsmore. Wouldn't shorter mandibles give bees more leverage, making it easier to damage the mite? And Jack Grimshaw had also said, wouldn't shorter mandibles provide more mechanical advantage?
Yeah, I think that's what we tend to draw the conclusion on. So with the a little bit smaller mandible, then the worker bee has a more flexibility to manipulate. So in addition, I didn't mention the mandible has a really wide variety of purpose for the colonies. They use mandibles to build cell, to feed the young, because there's a mandibular gland secrete. They can secrete the jelly into the brood, and they also use mandible to clean the cell, into yeah, to also defend the mite. So kind of think of, you have I try to think of a scissors example. You have a smaller scissor, maybe you can cut it better in a way, compared to a big scissor.
Yeah.
Then yeah, we also want to compare the body size. I'm trying to find a student to work on that project. It'll be interesting to see if their body size can change as well with the climate change, or with this, with the my population. Yeah.
Okay, great. We'll have one last question on that, and then there's a bunch of questions about the brood break. Maybe we can wrap that into one question. So Eddie Sillay said, what are the epigenetic mechanisms or environmental factors that drive the expression of grooming behavior and mite biting traits in Apis mellifera? And how might selective breeding or environmental manipulation optimize those behaviors for varroa mite resistance without compromising other colony traits such as productivity or overwintering survival?
Yeah, that's an excellent question. We have data now to compare the um RNA methylation. So that's one part of epigenetics between the high biting and low biting bees. We are just in the middle of data analysis, trying to get that question answered. In addition, I think there's not really much other research out there to compare the epigenetics of the grooming behavior. So our lab, definitely, I'd love to do more along that topic. And according to the environment, I think that question is a very, very good question. I think it's asking how is the environment, right? Change um, the behavior of the honeybees and how is that help their colony survival. So that's a very interesting question because we had to look at the on one side, if we look at the managed honeybee colonies, there is natural selection, of course, but there's also human selection. So, um, like like Professor Tom Seeley mentioned before, he published a paper, several papers and even book to talk about Darwin style of beekeeping. So I think what he's trying to advocate is to have a less of human selection on that part and kind of give more back to the natural selection part. And so when we look at all these different selection forces, and then we also look at the environmental changes now, we have climate change, we have temperature change. We're gonna experience more summer, wet weather. Like this summer, this spring, we had a horrible spring in Ohio, and it's not really a good fuel season as well because it rains a lot. And then it has only like a few dry days, and then they all suddenly swarm with all the cool weather. And then you look at the summer production with the hot weather. We had a drought like almost a whole 4 weeks straight in Ohio. There was no water. Then you can imagine what impact on the flowers, and then that will directly affect the bees. So we don't really have a good like honey harvest this year. But I can tell you probably this type of climate change, we're gonna see it more and more. So with all these environmental changes, in addition, yeah, so it will be interesting to see how that will affect the bee colonies. But for honeybee, it's one advantage is a lot of people can do splits in the summer. If they have strong colonies, they can always split and make into two strong colonies in that way. Yeah, so there are a lot of factors to consider there, but I would say combining all these different factors, a colony will always, try to involve in a way to benefit their survival better. So that because that's what the selection, yeah, it's going to select on. Yeah.
Right. And I'm going to let you make a choice about whether you want to answer this live, or maybe we can put a short paragraph together around the brood break that we could put with the recording. Your choice. So I'll just read you because there are a bunch of related kind of questions. So how long does a brood break have to be effective? Do you see any effect on winter bees in the fall? Does the lack of a brood break increase supersedure? When would you cage the queen in the Northeast, September or October? And breaking the brood cycle in the fall would limit production of fat winter bee population. So those are all the brood break questions that came in, and I'm happy if you want to address it, live now, or if you prefer to just put something together we can put with the recording, whichever you think would be best.
Yeah, so yeah, those are really good questions. I think for the mite control, we really want to target the highest peak season of the mite reproduction. So if you look at the, if you people do mite wash, you probably have some idea like when the mite gonna go crazy in the fall, and that's the time we try to target. So because I'm located at Ohio, we target pretty much the time of the year is September, because that's towards the end of the year. And I do realize that if we cage the queen, there's an impact on the brood to go over winter. So that's a very good question. We want to study that part as well. Just right now, we don't have data yet to show if we cage the bees, how do they go over winter? But as just from the common sense from Chinese beekeeping, I know it works because the beekeepers in China do use it. They typically do 3 weeks. So you can aim for the high peak of the mite production and just aim for that area, you know, left or right, but the peak time should be in the middle of the 3 weeks, and that will, yeah, like hopefully help to limit the female my reproduction. And yeah.
Great, lovely. Well, thank you very, very much. That's been absolutely fascinating. And I just have,
Yeah. Well, I just want to, maybe I'll add into one more point.
sure.
I'm not against chemical controlling the chemical management, but I want folks to be aware. In the United States, the Varroa, some of the population have resistance now. So you you if you use a chemical, I highly, highly recommend you to rotate them because the mice, yeah, will already have resistance now to the chemicals. And yet, then, if you want to, I would say the best way probably try to explore different ways, chemical way or non-chemical way to control, yeah, and not just only rely on one chemical. Yeah.
Great. Lovely. Thank you very much. So we have a few more things I just want to share with folks. We've done the poll. There we go. I'll flick through that. If you are interested in working with colleagues who are interested in pollinators and bees and the genetics that Dr. Lee Biley has been talking about, we have a Find a Colleague link on our website, primarily for people who are in the Northeast, but feel free to use it for other regions, I think. So you can put your link, your profile up there. And then also if you're looking for someone to work on a project with you, so you may have an apiary and you might be interested in working, for example, with Dr. Lee Biley as a site, then you can get in contact that way. So that's one of the things we do to try and connect people. If you're interested in being a peer reviewer on grant proposals for the IPM centers across the whole country, we have a link here that you can volunteer to be a peer reviewer. We're always looking for people who are growers, people who are in industry, and of course researchers to review our grant proposals. We have upcoming webinars. There's a research update conference on December 10th. It's actually fun. There's like 5-minute presentations, 3 slides, 5 minutes, and followed by a 10-minute Q&A for each half hour. So it's a great way of seeing what research people are working on right now. And then in February, we have got a pest management strategic plan and crop profile webinar and a weather app webinar also in February. A couple of people are coming to talk about different weather apps that are available. The recording of this will be available probably sometime next week. I'll send you a link to the recording, and anyone who's registered will get a copy to that link. I want to acknowledge that the Northeastern IPM Center is based at Cornell University in Ithaca, New York. Cornell University is located on the traditional homelands of the Gayokono, the Cayuga Nation. The Gayokono are members of the Haudenosaunee Confederacy, an alliance of 6 sovereign nations with a historic and contemporary presence on this land. The Confederacy precedes the establishment of Cornell University, New York State, and the United States of America. We acknowledge the painful history of Gayokono dispossession and honor the ongoing connection of the Gayokono people, past and present, to these lands and waters. This land acknowledgement has been reviewed and approved by the traditional Gayokono leadership. And I also want to acknowledge where we get our funding from, which is USDA NIFR. And this presentation would just not be possible without that grant. With that, I want to say thank you very, very much for this fascinating webinar and all the hours and hours and hours that have gone into making the presentation possible in so many ways. And I know there's been lots of great questions and lots of interest. So, and I know it'll be watched by thousands of people after this in the recording. So thank you very, very much for your time and your diligence and your expertise and your generosity of spirit in sharing it all with us. So thank you.
Yeah, thank you so much, Yana and David. I truly pleasure.
Great. Lovely. And with that, I will end the webinar. Thank you for attending.